Maxim Gilbert was an early method for DNA sequencing. Published in 1977, it uses radiation to label and chemicals to cleave, that is to cut, DNA for analysis. In DNA sequencing, we want to figure out what a particular piece or sequence of DNA reads. DNA is composed of 2 strands of nucleotides, which in turn are composed of 1 of 4 bases, in addition to a sugar and a phosphate group. The 4 bases are guanine, cytosine, adenine, and thymine. These can be abbreviated as G, C, A, and T, respectively. Each base has a complementary base pair, so if you know what one strand of DNA reads, you can quickly figure out what the other one does as well. The 5' and 3' labels mark directionality of the strands. The 2 strands of DNA are anti-parallel, so they run in opposite directions. 5' just refers to the carbon number along the sugar components of the nucleotides in a given strand. Thus, in sequencing, we are figuring out whether each position in the sequence contains a nucleotide corresponding to a G, C, A, or T. We will use as a simplified demonstration this 7 nucleotide stretch of DNA. Sequencing in practice can involve much longer sequences, thousands, even millions of nucleotides. DNA sequencing has a wide variety of uses, such as in evolutionary biology to see how organisms are related, in medicine to help doctors and patients assess genetic disease risk, in forensic identification, like at crime scenes, and in paternity testing. We begin the Maxim Gilbert method with double-stranded DNA, which is labeled radioactively at its 5' end. Then these double-stranded DNA are denatured or pulled apart via heat and/or chemicals, then purified to yield single-stranded DNA. These 5' radioactively labeled single-stranded DNA are then treated to 4 different chemical reactions. Each of these 4 chemical reactions randomly cleaves in a specific manner corresponding to nucleotides, creating fragments of different lengths, ranging from 1 nucleotide to its full length. 1 chemical to cleave at G, a second to cleave at A or G, a third for C, and a 4th at C or T. These fragments can then be subjected to electrophoresis in a polyacrylamide gel, separating fragments by size. Smaller fragments travel faster to the bottom. Courtesy of an audio radiograph and the radioactive labeling from earlier, we then get something akin to an X-ray of the polyacrylamide gel electrophoresis. We can recover the original sequence by reading from bottom to top. Each of these columns pertains to one of the 4 chemical reactions. For reactions that cleave at either of 2 nucleotides, identification of nucleotides can be resolved by looking at another column. The band at the bottom occurs in the A plus G column, but with no band in the G column, so we know the first position is an A. The next one is in the C plus T column, but there is no band in the C column, so we know it's a T. The third one fills both the C and the C plus T column, so it's a C. Same idea for the 4th position, it's a G, with both the G and A plus G columns filled. Then another T, a G, and one last C to wrap up our sequence. Maxim Gilbert was a first-generation sequencing method and is no longer commonly used. It was initially more popular than the Sanger sequencing method, a fellow first-generation method, but Maxim Gilbert eventually lost out in favorability to Sanger sequencing. With improvements in the Sanger method, Sanger was deemed faster and more scalable. Like Maxim Gilbert, the original Sanger method used radioactively labeled DNA. Later enhancements to the Sanger method transitioned to fluorescently labeled DNA instead of radioactively labeled DNA. This reduced the reactions needed down to 1 rather than 4. However, since then there have been so-called next-generation sequencing methods, and subsequently third-generation sequencing methods, offering improved speed, efficiency, and cost.